Okay, well, welcome everybody and thank you for joining. This is our second innovation workshop of day 2. So today I'm delighted to be joined by 4 of our One Nucleus members who will be showcasing their work and various contributions to the industry. So just a few housekeeping points before we get going. If you can keep your microphones on mute and your cameras off throughout the presentations, that would be fantastic. And if you do have any questions and comments, please feel free to put them in the chat box. If we don't get around to picking them up today, we'll make sure that we connect the dots after the session, and that will be absolutely fine. So I think that is it from me. I'm going to hand you over to our first presenter, Victor Francis, who is the Director of Business Development at Diversa Technologies.
Thank you, Jazz, and thank you everyone for attending the innovation workshop. I'm going to share my screen. Just let me know if you can see it.
Yeah, looks good.
Yeah, great. Well, thanks again. We're very excited to be here. We just joined One Nucleus around 6 weeks ago. We're a young biotech company from Spain. And we're very thrilled to share a little bit about Diversa's story and about our technology. And we look forward to meeting all of you on Thursday if you happen to be in Cambridge in person. A little bit about Diversa. As I mentioned, we're a Spanish biotech company. We're based in Santiago de Compostela, which is northwestern Spain, where the Camino de Santiago ends, if you've heard about that. We're a multidisciplinary team with pharmacists, biochemists, and medical doctors. The co-founders have Maria Lafuente, our CEO, has more than 20 years experience in nanotechnology. And this is actually how the whole idea of Diversa started. She was getting requests after every talk that she gave by peers in the drug discovery world about, hey, could we encapsulate some of these molecules using your systems? And that's where Diversa started. So Abby's our CEO, and she's a co-inventor of the patent. And Rafa Lopez is our medical advisor. He's also the head of the oncology department at the University Hospital in Santiago. So he keeps us connected to the clinic, which is very important for us. So we established in January 2022. We got pre-seed funding from the Galician government, which is the regional government in Galicia, northwestern Spain. We are 7 employees at the moment, and we have 1 patent, which is the cornerstone for the whole project and the company. And we have commercial activities in the European Union and the UK. We are very close to closing an investor seed funding round. We can talk about that as well in person on Thursday. The team is very multidisciplinary, as I said. The focus is in the lab. We have 4 people at the moment running all the projects and developing the technology further. And we are actually looking for still a researcher position. So we're going to post that on Helix website. But if you know of someone that would like to move to sunny Spain, just let me know as well, and we'll connect. The idea of Diversa, again, started with realizing that almost 97% of new molecules that get discovered in basic labs around the world never make it to the clinic, either because their solubility is poor, or they get degraded when they're inside the body, or they're not able to enter the inside of the cell, which is where 3 out of 4 new molecules have their therapeutic target. With our technology at Diversa, we wanted to give new opportunities to at least some of these molecules to make it from bench to bedside, from the basic research to the clinic. So we developed this technology, which is based in lipid nanoemulsions, LNEs, we call them, in comparison to LNPs. They're biocompatible and biodegradable, and they can be used in pharmaceutical drug development, but also for nutraceutical and cosmetic applications. Our technology has been designed with translatability and simplicity in mind, and this slide is just to represent complex nanosystems, say LNPs, liposomes, lipidic bilayers, or several encapsulations, whereas our nanosystems are lipidic monolayers, micelles, basically fat drops, and they're cheaper to produce, easier to scale up, and the composition is simpler. The two main components are two naturally occurring lipids, and this is the patent information that you can find online, which are sphingomyelin and vitamin E. They're both naturally present in the body, and they're also approved as excipients in other drugs that are commercially available in both by the FDA and the EMA. So the patent was published in 2019, so it's quite fresh, and the main focus of the company is to develop new drug delivery technologies based on this patent. So a little bit about the science behind the technology. Two of the main important physical-chemical characteristics that we measure in our nanosystems are size, diameter, and electrical charge. You can see here in the top left graph, our nanosystems are usually on the lower nanometric scale between 100 and 200 nanometers. This has some advantages when using these systems in vivo in humans, for example, using an intravenous administration route, because the smaller these nanosystems are, the less risk to cause some blood clots, for example. The other important factor is the charge, and that would be the top right graph. It's called zeta potential, but it's basically a representation of the electrical charge of these systems. And the closer they are to zero, neutral charge, the less off-target effects or toxicity associated to these nanosystems, because they won't be able to bind to the wrong cell type or different molecules once they're inside the body. We also looked at stability. We first did the top left graph is ICH normalized accelerated stability studies. This is just to see the shelf life of these lipid nanoemulsions. And we could see 6 months, which equate to 2 years shelf storage for these nanosystems. We also did very shorter times in comparison using different buffers and cell culture media. We supplemented with or without FPS, and we didn't see any change in the stability of these systems. The FPS part is very important because one of the key characteristics of our nanosystems is that they do not have cationic lipids. And these cationic lipids, that are used to bind the cargo molecule using electrostatic charges, FPS masks all these interactions by charge. So we're using some covalent bindings, and FPS doesn't really affect it. So it's an advantage if the researcher can use or not use FPS when using our technology. Then we looked at some biological fluids just to see how well our nanosystems would fare using different administration routes. So we checked for human plasma, tear fluid for ophthalmic applications, synovial fluid for intraarticular, and gastric and intestinal for oral administration. And I will show some biodistribution data later on. We also looked at biocompatibility, and we did that in different cell lines. Here, we're showing fibroblasts and hepatic HepG2 cells with increasing concentrations of the nanosystems and a very high dose of 10 mg per ml. We didn't see reduced viability of these cells. Also, when we tried these in mice with two very high doses of 30 and 60 milligrams per kilo at different days, we didn't observe a change in the weight of the animals or a change in the behavior of these animals. And of course, none of the animals died after the treatment. We also checked the toxicity using zebrafish embryos, which is this image on the bottom right, and the graph on the bottom left. And you can see that control and increasing concentrations of the nanosystems didn't really affect the viability of these animals. Then we have a very versatile system in that we combine small hydrophobic drugs, peptides, proteins, and oligonucleotides, and all of them with high association efficiencies. Top right is a very crude representation of our nanosystems, but as I mentioned before, they're basically fat droplets with an hydrophobic core and hydrophilic groups of these sphingolipids and vitamin E on the outside. So most hydrophilic molecules will bind to the outside, so they'll be associated to our nanosystem, whereas hydrophobic molecules will go to the inside. And therefore, in general, we have a higher loading capacity for hydrophobic molecules. Nevertheless, we can use some surfactants to recover a hydrophilic molecule and then encapsulate it inside. But these, in comparison, for example, to liposomes, where you have a lipidic bilayer, our loading capacity for hydrophobic molecules is much higher. Then we look you not only want these molecules to get associated to the nanosystems, you want them released into the cell. So we looked at cell internalization. The graph on the top left is a representation of the image below of fluorescence microscopy, and we did a time course to see how fast our nanosystems labeled with a green fluorochrome would get into the cells. And after 60 minutes, most of these cells, in this case fibroblasts, had the nanosystems inside. We looked at a battery of different cells, and just to show for all of them, after 60 minutes, they were 100% internalized, even when doing primary cultures such as astrocytes. The previous slide was with empty nanosystems. When we deliver a cargo molecule, we can see the effects of using Diversa Technologies. So on the left is a peptide. In this case, the peptide is labeled with green fluorochrome. And you can see that when we incubate the free peptide with these fibroblasts and we wash the media, there's no peptide or barely any inside these fibroblasts. When the free peptide, the peptide is associated to DIFTEC, which is the commercial name for our nanosystems, you can see that it enters the cells and it colocalizes with the red marker, which is rhodamine for the cytoskeleton of these cells. The same goes on the right for a protein, beta-galactosidase, which is 540 kilodaltons, pretty big. And you can see on its own, it cannot enter the cells. When it's associated to DIFTEC, it enters the cells, plus it's active. So it's not just inside the cell. The blue color is caused by a substrate of beta-galactosidase that turns blue once it's enzymatically acted on by beta-galactosidase. This one is just to show the biocompatibility aspect of our technology. The first two images, left and center, are the same ones as in the previous slide. Free beta-gal doesn't enter the cells. Associated to DIFTEC, it goes into the cells. And then we used a competitor. In this case, it's a commercially available liposome delivery system. You can see that beta-gal goes into the cells, but you can also see that the cells don't look that good. And there's a lot of cellular debris, which is an indicator of cellular death. And this is only after 2 hours in vitro. So if you're thinking of using this delivery system for an in vivo therapy, you understand my point. So when we looked at systemic biodistribution, we did this with empty nanosystems labeled with gallium-68, which is a radioisotope. And we did PET scan in mice. So the image on the left are these mice. The yellow blob is the liver. And you can see in the graph in the center that most of the nanosystems localized to the liver, kidneys, and spleen. This is normal for lipid-based drug delivery systems. What we were thrilled to see is that we also saw quite a lot of incorporation to the lungs. Treating lung-based diseases, lung cancer, and also in the blood. And we can see this was an intravenous injection through the tail vein. When on the graph on the right, when we compare intraperitoneal and intravenous injection, you can see that we had the same liver, lungs, and bone marrow, which is in the blood. So we also checked for intraperitoneal administration, in this case with the nanosystems labeled in green with our green fluorochrome, and we did ex vivo localization or study of the organs. You can see that both at 8 hours and 24 hours, we could see in the liver, but also interestingly in this tumor, because this is a breast cancer mice model, and you can see that these tumor cells have soaked up of the nanosystems, probably because it's more active metabolically. So to the business model, we offer our technology through different ways. The first one is in the form of kits, ready-to-go kits that any lab research center can buy and test if they can encapsulate and reformulate their molecule of interest. Then we do co-development agreements, mostly geared towards biotech and pharma companies, where they send us their molecule, we reformulate it, and then we send it back and they can test it side by side. And then we do our own R&D. So the kits, they're actually sold in the UK already through our distribution partner, which is Tebo Bio, a French scientific distributor. We have kits for small molecule peptide and protein at the moment, and we have both versions with or without the green fluorochrome, so that you can do localization studies and follow up. All these advantages with previous drug delivery technologies I've mentioned, we're independent of molecular weight and charge, independent of FPS, which is very important if your cells need FPS to survive in vitro. We do not use cationic lipids, which gives us this advantage in biocompatibility. We're versatile in that we combine different types of molecules. And on top of that, we offer the customized service, which looks like a service contract first. There's a material transfer from the biotech pharma company to us. We also work with academic labs, and we can adapt to the different situations. We reformulate, we give the testable prototypes back, and the ones that perform the best, if the customer wants to bring them and produce them commercially, we can then enter a license agreement for the specific indication and molecule. We do have CDMO partners that can produce at scale in GMP conditions. And we do our own R&D because we don't want to be only a platform technology company, but we want to be able to further that technology and bring new drugs to the market based on our technology. This is the timeline. We incorporated in January 2022. We have the first co-development agreements. We're commercializing the kits, and we're looking to expand into the UK, US, Japan, increase our commercial activities. And yeah, so I'll be happy to meet all of you on Thursday in person. And we're looking for projects either for our kits or our co-development agreements. And any proof of concept projects we can adapt to that if it's very interesting. And yeah, also looking for public and private funding together. And that's all.
Thank you very much, Victor. So can I please have Campbell Bunce next from Abzena? Wonderful.
Let me share a screen. Yeah.
Great.
Can you see the screen?
Yeah, looks good.
Otherwise, my slide good. Okay, well, thank you to Tony and the One Nucleus team for giving me this opportunity to talk at this year's On Helix event about one of my favorite subjects, which is Abzena. I'm the CSO and site head of Abzena Cambridge facility based on the Babraham Research Campus. And in this short presentation, I will cover who we are, what we do, how we do it, and why we do what we do. So Abzena is a service provider and leading partner research organization supporting integrated discovery, development and manufacture of biologics and bioconjugates. We use the term partner as we work in a highly collaborative way to take our customers' projects from concept to patients. And we work across 4 locations. One of them is here in the UK, in Cambridge, where we focus on discovery, design to lead selection, and cell line development. In San Diego, California, we deliver process development and GMP manufacture of biologics for clinical testing. In Bristol and Pennsylvania, we deliver synthetic chemistry, process development, GMP manufacture of linker payloads and bioconjugates. And in Sanford, in North Carolina, this is a new facility that's opening this year, and we will deliver phase 3 and commercial drug substance and drug product from that new site. So we offer a range of services either as an integrated program or individually across all stages of development for large molecules in the form of antibodies, fusion molecules, recombinant proteins, small molecules in the context of complex compounds, particularly linker payloads, and we have the ability to handle large quantities of highly potent APIs. And these come together to support bioconjugate and ADC design, development, and manufacture. And in this sort of resurgent era of vaccines, we have adapted some of our core services, such as protein expression and production, bioconjugation, proteomics to support vaccine design and development too. Our focus is very much on the critical development path, getting our partners from where they join us in that path to IND and beyond, ensuring the best quality within a rapid time frame. And we do this by employing a number of our workflows and platforms. For example, we have a hybridoma antibody discovery platform, but we'll soon be introducing B-cell antibody discovery, leveraging the Berkeley Lights beacon system to shorten the time frame from immunization to antibodies to 14 weeks. This accelerated workflow includes a rapid, high-throughput expression platform to generate more data across more leads in a quicker time frame to select the best candidate to take forward into development. We also have been continuously improving our cell line development platform. This includes the generation of a new CHO-K1 host line, and the use of another local group, Sphere Fluidics, cytomine system to reduce the timeline from DNA to RCB to 15 weeks. And one of the great advantages of our CHO-K1 platform is that it produces a lot of material at the stable pool stage. And this supports the production of an early and substantial batch of material. For example, we can produce 5-10 grams of purified antibody within 8 weeks, which can be used to take a number of downstream activities off the critical path, such as small-scale process development, for example, purification and assembly processes for ADCs or bispecifics, analytical method development, preliminary stability studies, formulation development, and even in vivo studies, including dose ranging and non-GMP tox. An aspect of drug development built into our sort of ethos at Abzena as a solution provider is developability. This concept underpins successful drug development by focusing on key questions early in the development journey. So can we make it? Can we make it to the scale and form needed to dose patients? Does it work? Does it do what we want it to do, expect it to do? And is it safe? Does it work within the accepted safety parameters for the disease we are treating? And we adopt a holistic approach, multi-parametric approach for this, evaluating specificity and functionality, immunogenicity and safety, and manufacturability, with additional considerations for ADC and bioconjugates. And through this process, we can identify risk factors early in development. And this allows scope to apply design alterations to reduce or manage these liabilities, and consequently reducing the likelihood of major issues arising at a later, much more expensive phase of development. And this ultimately ensures that we do take the best drug candidate forward to manufacture. Underpinning our developability approach is extensive analytical and bioassay expertise and capabilities, as well as the scientific and technical skills to design out or manage liabilities through protein and chemical engineering and formulation development. And this slide gives some indication of the breadth and depth of analysis that we apply to drug candidates to identify, where necessary, modify the best lead to take forward. So we use a combination of in silico, in vitro, ex vivo assays, alongside an array of biophys and phys-chem analytics to meet this ultimate goal. ADCs and novel bioconjugate drugs are worth special mention, as they're proving to be a major area of success in the drug development sector. And Abzena are world experts in this field with a huge amount of experience in the design, scale up and manufacture of bioconjugates and ADCs. We have an extensive toolbox to work from to create a wide range of bioconjugates, including use of our own proprietary payloads and linkers, which we offer license-free to our partners. And through a matrix evaluation process, we can assess combinations of the key ADC components, so the targeting proteins, the linker, and the payloads. We can design and select the best candidate to progress to process development and manufacture. And once we've designed and selected the best lead candidate and generated the production cell line, we transfer that to our versatile manufacture capabilities, where we utilize a range of single-use bioreactors from 50 to 2,000 litre scale, depending on the material requirements. And this is integrated with extensive downstream process capabilities to deliver the GMP materials and product for our partners. So I'd like to end with our mission statement, your vision, our mission for patients. Our ultimate goal or ultimate aim really is to add value for all stakeholders towards the improvement of human health. So we strive to give the drug the best possible chance of success for our drug developer partners. And this supports a smoother approval process with the regulators, as well as indirectly supporting the expectations of investors who of course fund their partners and are looking for a return on their investment. And ultimately, we are adding value for patients by contributing to successful drug discovery, design, development, manufacture, and the generation of new and improved drugs to better treat disease and to improve human health. So I appreciate that was quite succinct. I've probably made up a little bit of time, I hope. Been a bit paranoid about taking my fellow speakers' time up, but I'm happy to take questions if we have any time. Otherwise, please get in touch if you'd like to discuss how we can help you develop your drug.
Thank you very much. We do actually have one question, and I'm sure we can squeeze it in quickly if you're happy to.
Yeah, sure.
So that is from Marcel, and it is, thank you, Marcel. Is the Abzena CHO cell line suitable for clinical trial manufacture, derivation history, etc.?
Yes, no, it's been purpose-built exactly as a production line. So we generate research cell banks and then pass them over to our manufacturing facility in San Diego to make master cell bank for manufacture of GMP product. The fact that we can use it early on to make quite a bit of material at bulk pool stage is a bit of a side aspect, but it's principally for a production cell line for GMP material.
Thank you very much. Okay, I'm pleased to invite our next presenter, Mike Sheldon, who is the Senior Director of Scientific Affairs for Sampled.
Okay, thank you. Let me make sure I'm sharing my screen.
Wonderful. Have you got your tablet here? Wonderful.
Okay, can you see my screen?
Yeah, that's great. Thanks, Mike.
Okay, brilliant. And I will get started in the interest of time. Good morning and good afternoon to colleagues on either side of the Atlantic. My name is Mike Sheldon, and I'm Senior Director of Scientific Affairs at Sampled, a company in central New Jersey in the US. Our new 200,000 square foot laboratory facility is pictured here, and I'm speaking to you from New Jersey today. From its origins supporting the US National Institute of Health to an extensive portfolio of pharma and biotech clients in the present day, our company has been a prominent participant in the biobanking and sample processing community for more than 25 years. And I'm pleased to say we've been a member of the One Nucleus community for more than a year now. Today, I'll present an overview of our service offerings with emphasis on our innovative approach to sample processing using a smart laboratory model. I'll also introduce our newest laboratory facility in the UK and touch on the processes it adds to the sampled template. Okay. Slides are not advancing, so there we go. Computer's waking up this morning. The Sampled Smart Lab model is designed to provide comprehensive solutions with maximum options and flexibility in support of customers in a diverse array of fields. So what does SMART stand for? Well, storage capabilities include monitored biobanking and retrieval of stored samples, as well as ICH stability storage for pharma and medical devices. Management, we manage projects at the level of the study all the way down to the individual sample, working with clients from the very beginning, even before their trials or projects start, with dedicated project managers assigned to each client. All methods of nucleic acid extraction, quality control, and analysis are available, including genomics, transcriptomics, and epigenomic arrays and sequencing. I'll have more to say about those technologies in a moment. Our research teams offer comprehensive services in cell and stem cell culture, pathology, and assay development. Dedicated sample management and transport teams are responsible for cold chain logistics that are compliant with the requirements of all major international regulatory bodies. Sample collection, kitting, and logistics services are offered that support the proper collection, transport, and processing of any biospecimen that can be collected from a human. To this end, blood, saliva, breast milk, feces, urine, toenails, you name it, are collected in kits provided by Sampled for our clients and processed in our laboratory as well as in others. Each step of this process, from storage, management, analysis, research to transportation, is customizable and safeguarded by standard operating procedures that are rigorously adhered to. A bit about our history. Our involvement in the biorepository and sample processing field is characterized by a long history of innovation. From our origins, on the left here, as a nonprofit academically based biorepository formed in 1996 and known as RUCDR Infinite Biologics, affiliated with the State University of New Jersey, Rutgers University. Our earliest efforts were focused on support for several agencies of NIH, and this continues to this day, serving as the biorepository and sample processing center for NIMH and NIDA, to name two. In the years that have followed, our portfolio has grown to include numerous clients in the commercial sector, both pharma and biotech. I'll touch back on those later in the talk. Now in 2020, the pandemic entered all of our lives, and we rose to the challenges presented by the pandemic by developing the first saliva-based COVID qPCR test to receive FDA emergency use authorization. This innovation, coupled with our expertise in scaling high throughput operations, has enabled us to test more than 12 million samples to date. In August of 2020, we made the transition from a nonprofit to become a commercial entity known as Infinity Biologics. As IBX, our commitment to innovation gained momentum, and 2021 was a year of significant milestones for our company, some of which are highlighted here. As a natural extension of our large effort in COVID testing, the CDC awarded IBX one of the 6 contracts in the US to sequence COVID-19 positive samples as part of its variant surveillance program. We've also gone on to develop and bring to market 2 polygenic risk score tests for breast cancer and ovarian cancer risk and Alzheimer's disease. We later in the year welcomed UK-based Roylance Pharma to the IBX family. Roylance brings years of experience in first-class biobanking and storage, expanding our footprint overseas. And I'll have more to say about them in a moment. In April of this year, we rebranded and our name was changed to Sampled. But while the name changes, our commitment to quality and our track record of service remain strong as ever. This slide is to give you a little bit of a snapshot to give you an appreciation of the scale of our operations. Now at Sampled, we define our core projects as those that are distinct and separate from COVID processing. These are projects with clients we worked with well before COVID and will continue to work with well after the pandemic has finally ended. This slide presents an annual snapshot based on last year of our processing of a variety of different biosamples for our core projects. As you can see, the majority of samples received were blood and blood products destined for blood fractionation, isolation of PBMCs in plasma, as well as isolation of nucleic acids such as DNA or RNA. We also received a large number of saliva and other body fluids as well. Of the over 500,000 samples highlighted in the middle of the graph that we we processed and stored 194,000 and submitted 312,000 others directly for processing. Some of the highlights to be gleaned from this slide are the more than 100,000 samples subjected to blood fractionation and over 300,000 that underwent analytical services, including nucleic acid samples extracted both in our labs and by external labs. And the breakdown of these services is shown here, including microarrays, sequencing, and qPCR. At Sampled, we've built the infrastructure and workforce needed to support College of American Pathologists compliant storage of biomaterials at any required regulated temperature, ranging from ambient to liquid nitrogen. And this slide summarizes all of the breakdowns of those temperature ranges as well as biomaterials. The variety of biomaterials stored in our biobank reflects the wide variety of sample processing services we offer. Again, anything that can be collected from a human, including blood fractionation, cell line derivation, nucleic acid extraction, quality control, and tissue processing. We've also, as reflected on this slide, developed some innovative approaches to the analysis of human stool samples for microbiomic studies, as well as semen for the characterization of sperm health and motility. Our laboratory in Glasgow, formerly Roylance Pharma, brings years of world-class experience in the areas of stability and controlled storage of medical devices, compounds, and biomaterials to the Sampled family, as depicted on this slide. Unfortunately, time doesn't permit me to go into depth on these, but I would encourage you to visit the Sampled team at stand number 4 on Thursday at the On Helix meeting in-person event. I regret not being there in person myself, but our team will be happy to answer any and all questions. The maintenance of the highest standards in sample integrity starts with quality. Quality is at the center of everything we do. Our quality team oversees all aspects of SOP and document control, technician training and competency, and regulatory compliance. Sampled is proud to have been one of the first laboratories in the world to receive accreditation through the CAP, College of American Pathologists Biorepository Program, as well as maintain best practices consistent with all of the international organizations and licensing bodies shown here. Consistent with this, all freezers and tanks are independently alarmed and monitored 24/7, 365 with dedicated backup power generators. All collection vessels in sampled collection kits are pre-barcoded to ensure an unbroken chain of custody from the subject to the labs to recipients for research. On receipt, samples are inspected and imaged during accessioning, and their processing tracked in real time in our LIMS using those same barcodes. Assays are performed on all biospecimens processed at sampled to assess measures of analytical and functional quality control. Advances in molecular analyses based on high throughput technologies can contribute to a more accurate classification of disease and as tools to better predict both the disease course and efficacy of targeted therapies. Our clients, large and small, we work with pharma all the way down to individual investigators, have access to an industry-leading variety of genomics technologies in our labs, ranging from genotyping and transcriptome arrays to all major platforms for next generation, second and third generation sequencing. I'll tell a bit more about our single cell work as well. Epigenetic analyses on methylation arrays or methyl-seq and large-scale qPCR studies. Many of these genomic services are available as either research use only or clinically validated assays in our CLIA laboratory. This slide is a snapshot of genomic analyses performed in 2021. And you can see the scale of operations, including a very large effort in sequencing. As a demonstration of our sequencing capabilities, our participation in the CDC COVID variant surveillance program is an illustration of our capabilities as well as a case study in our ability to leverage our experience in high throughput sample processing to scale up operations in response to a crisis. Samples that are identified as COVID positive and meet maximum CT criteria in our PCR testing lab are submitted for COVID-19 sequencing using the Illumina platform and COVID-seq kit. We've developed the capacity to sequence as many as 6,000 samples per week with an average of 3,000 per week since April. This graph depicts the dominant CDC strain variants of concern identified in our pipeline over the course of 11 months, with the initial surge of Alpha, the blue graph, being supplanted in succession by Delta in July of 2021 and Omicron in January of 2022. These efforts resulted in the publication of a paper in BMC Infectious Diseases in April of this year. Sampled has made a significant commitment to the implementation of multiomics and single cell technologies as well. We are a certified service provider for the 10x Genomics Chromium single cell RNA-seq technology and are in the process of validating their Visium spatial transcriptomic platform for incorporation into our service menu as well. This powerful technology will enable our clients to interrogate gene expression across the geography of a tissue section, allowing inferences to be made about the relationship between expression, tissue milieu, cell morphology, and protein expression using immunohistochemical markers, for example. We're also excited to be adding Olink's platform to enable interrogation of protein expression in a variety of samples and projects. And I'd be happy to discuss that on a separate conversation. To give you an illustration of a specific field that benefits from the services that we offer, and again, we offer subsets of the smart lab approach to a variety of clients, as well as the complete package. And these are offered to companies that either wish to outsource these capabilities altogether or need support as their demand exceeds their internal capacity over time. So the illustration on this slide is for the cancer cell therapy field. Current sampled capabilities of relevance to this field include nucleic acid extraction in QC using a variety of chemistries, next-gen sequencing, as I said before, RUO or clinical, and digital droplet PCR for quantitation of vector copy number, minimal residual disease, potential pathogens, etc. We also offer a wide variety of histology services as shown on this slide. Our cell and stem cell labs have a variety of different services to offer, including the isolation and culture of primary cells like PBMCs and fibroblasts, the derivation of induced pluripotent stem cells, and maintenance of master and working cell banks as illustrated here. We have established and continue to expand a global presence. In blue are our main laboratory processing centers in New Jersey and Minnesota in the US, as well as in the UK. And we have partner labs in other countries around the world. And lastly, I just wanted to add that we're very excited about our Glasgow lab increasing its capabilities in a variety of areas as shown on this slide, using SOPs and processes that are identical to those in the United States to maintain consistency across the company. List of our clients and partners and our leadership team shown here, without whom none of this would be possible. And I thank you for your attention.
Thank you very much, Mike. That was brilliant. So this now brings us to our final presenter, and I'm delighted to welcome Amanda Woodruff, who is the Senior Vice President for the Precision of Medicine.
All right, thank you. I'm just going to share my screen, I hope.
Excellent. Are you able to turn your camera on,
Yep.
Amanda?
Sorry?
Are you able to turn your camera on?
Oh, sorry, did I not do that? Sorry. Bear with me a sec. Oh, hang on a minute. Start video. There we go. There we are. Okay,
Wonderful. Thank you.
great. Can everyone see that?
Yeah.
Okay, great. Well, again, last but not least, but I'd like to thank One Nucleus for the opportunity to come and have a talk to you today about precision for medicine. As mentioned, I'm the site head and Senior Vice President. I run Precision for Medicine's lab that's based in the UK. Unlike our first speaker and Mike, we've been members of One Nucleus for a little bit longer than sort of 6 months to a year. I think we've been members for over 20 years now. So for those of you that are as old as me and remember Irby, which was before One Nucleus, yeah, we've been One Nucleus members for a very long time. So with that, I'm gonna carry on, and I'm gonna see if I can get rid of that off my screen. So there we go. Okay. So for those of you that aren't familiar with Precision for Medicine, we are a full service CRO, and we're really a biomarker driven, as the name suggests, precision medicine. We're really biomarker driven clinical development provider. So, and that's really anything and everything from the clinical trials themselves, the design, the execution of those clinical trials from our clinical teams, through to the provision of specialty lab services, also central lab services. Mike touched on that during his presentation, the kitting, logistics, sample protection, getting the samples from the clinical sites to the analytical labs in the best possible quality to generate good downstream data, all the way through to our proprietary biomarker informatics platform and data harmonization. So we have a platform called Quartz Bio, and that's really built to look at integrating data from all types of sources, whether that's from patients, electronic health records, through to the sample data itself, through to the assay data, and all of that needs to be consolidated and integrated so that our sponsors can actually get the maximum value out of that. And then last but not least, we take that process all the way through from developing those specialty assays, and where appropriate, we're actually supporting the development of companion diagnostics. So we have a really strong internal regulatory team as well. So really, we are a kind of holistic, precision medicine driven clinical trials company, CRO. Okay. So, and we are global as well. So we've been in business for over 20 years conducting clinical trials globally. So the map here really just indicates the dark blue areas are where we have kind of clinical reach, so where we're actually establishing and managing clinical trials. We have a number of our own wholly owned specialty labs, of which my lab in Royston in the UK is one, and they're marked in the green dots. So in the USA, we have 5 precision labs in Massachusetts, Maryland, North Carolina, Texas, and one over on the West Coast in California. And we have 2 labs in the kind of UK European footprint. So we have our lab in the UK in Royston, which is just south of Cambridge, and we also have a lab in Berlin in Germany. So Mike very kindly touched on some of the themes I was going to talk about today for precision as well, but sample processing is really critically important. So clinical trial sample processing, particularly when you're looking at PBMC isolation from blood, oftentimes that's mission critical to have that isolation done within, say, 24 hours. So Precision has actually set up a number of partner labs. So these are not labs that are owned by Precision, but they are trained and operate to our SOPs. And we've dotted these around the globe, and you can see these as the blue dots on the map. So we have partner processing labs, and again, this is primarily, not exclusively, but primarily for PBMC isolations. So we have labs in Singapore, Australia, South Africa, and we have a couple of additional sites across Europe as well, including Spain and Austria. And then last but not least, the kind of gold coloured dots, these represent where Precision has sort of offices or legal entities across the globe as well, and these really are in support of our clinical teams. So we have CRAs and various clinical entities operating across the world as well. So what I'm going to talk about today is the specialty labs part of Precision. So this is where my lab fits in. And this slide here is really just to indicate that we have a very broad approach to being able to interrogate analytes in biofluids, tissues, etc. So I'm not going to go through this in a huge amount of detail. I just wanted to kind of put it up there just really to demonstrate our broad capabilities. So we can interrogate anything from sort of nucleic acids through to measurement of proteins. So we do a lot of cytokine analysis, so looking at analytes in serum, for example. A really strong focus area for Precision is flow cytometry. This is probably what the Precision labs were primarily known for a few years ago. So this is a really big area for us, and we now actually have flow cytometry capabilities that can go up to 30+ color for flow as well, usually for research use only. But these are assays that are developed to support sort of big investigative studies. And then last but not least, and really what I'm going to talk about a little bit later on, is tissue-based analysis as well. So we do have a lot of expertise in immunohistochemistry. So really this slide is there to kind of give you a snapshot of capabilities. The strategy at Precision is really to enable and to mirror the capabilities that we have to support our US sponsors running US-based clinical trials. So all of our labs in the US, and we have mirrored those capabilities and those platforms and those technologies across our 2 labs in UK, Europe as well. We support clinical trials primarily. I'm going to talk a little bit about preclinical work as well in a moment. But obviously the labs all have, across the Precision network, all have the appropriate level of regulatory compliance. So CLIA, ISO, various different ISOs depending on what that particular lab is actually delivering. So our companion diagnostics development lab based in the US is ISO 13485, and a number of our labs also operate to GXP as well. Okay, so focusing now just on the UK lab, say we've been around for a very long time, been members of One Nucleus IRBI for over 20 years. So the lab here in the UK has been operating for 26 years, and our background is in human tissue-based research. So primarily we started at the discovery end of the business, really looking at sort of target and biomarker validation. And as we have developed a lot of assays and we've evolved as an organization, we had started to kind of take ourselves from that kind of late stage preclinical all the way through to being able to then develop the assays and further validate those assays that we've developed preclinically to be able to support clinical trial assays. So our background is really in that kind of sweet spot of human tissue based and human biology based target and biomarker validation. So our core activities have really focused around analysing targets and biomarkers in human disease tissue cohorts using either immunohistochemistry or qRT-PCR or in situ. We've done a lot of work in the past in the development and application of program-specific tissue microarrays as well. So a kind of high throughput platform for bringing together big sets of data. So we can create TMAs that are very specific to a particular program of work, so in specific disease areas, and then use those TMAs as a tool to interrogate the markers that we're looking at, be that by IHC or ISH, for example. We have also generated many years ago now, and it's still data that we produce today for our clients. We have a qRT-PCR based whole body map, which is essentially gives you a kind of snapshot of your target of interest expression across a whole human body. And on the sort of therapeutic effect side, so we've talked a lot about targets when we actually come on to talking about molecules. So therapeutic effect and therapeutic safety, our sweet spot here is really designing and developing fit for purpose, so often quite customized human primary cell-based assays to support mechanism of action, and to look at, you know, the potential for certain biomarkers and, you know, the relevance of those in a kind of organotypic human primary cell-based system. So although our focus over the last 26 years has really been around human-based biology, the translation of target and biomarker expression between species is really critical as well. So we do also support quite a lot of you know, preclinical species work as well, and particularly in the areas of cell and gene therapy and some of the immuno-oncology areas. You know, that also includes non-human primate work. So I'm going to talk a little bit about probably what is our core area of expertise in the UK lab. And as I say, these capabilities are mirrored in some of our US labs as well. But the UK lab really brings to the party our expertise in immunohistochemistry. So this is really all about looking at target and biomarkers in tissues and being able to characterize the tissue microenvironment and changes in that tissue microenvironment in response to drug treatment. So IHC all starts with finding good antibodies. I know Abzena are at the conference, and you know, they're one of the ones out there that produce very good antibodies, but unfortunately not all antibodies are great. So the IHC has to start with a thorough validation of the antibodies, development of your IHC assay, and optimization of that assay. And then really that goes down one of three routes really. So we can use those immunohistochemistry assays to support target validation. So maybe looking across and surveying across disease areas or disease indications to see where that target is highly expressed. The second route is to go down GLP tissue cross-reactivity, which is an IHC technique for evaluating biotherapeutics. And I'm going to come on and talk about that in a moment. And then last but not least, once your assay is developed and optimized, we were able to take that through the appropriate level of assay qualification and validation, and actually then be able to use those assays to support analysis of clinical trial samples. And we operate in the UK to GCLP. We're also a GLP laboratory, as you can probably tell from the middle grey box there. So again, really the message here is that we've been doing immunohistochemistry and development of robust IHC assays for many years. We're a really well-equipped lab. We have all the industry standard autostainer platforms. We scan all of our chromogenic IHC using the Perio AT2. And for multiplex immunohistochemistry, which I'll come on to talk about in a moment, we use the Vectra-Polaris multispectral imaging analysis system. Data analysis is then either done through our pathology team or using specific software from Halo in particular, or actually for the Akoya, the Informed Tissue Finder, which is built into the Vectra-Polaris machine. Because we've been doing this for a long time, we've got a lot of expertise in actually developing these assays. We've got a lot of experience at working on some quite challenging targets. And I'm going to come on and talk a little bit in a minute about non-traditional binding molecules as well. And there I'm talking more about some of the actual therapeutics that people are developing as drugs destined for clinical administration. And as a consequence of having been around for a wee while, we've got a lot of work in assay protocols for a lot of different markers. And whilst Precision's focus is really in that kind of immune-oncology, autoimmune disease, cell and gene therapy, rare disease, our history as an organisation in Royston here, we've worked across many different disease areas. So we do have a breadth, which is actually really useful for our continued support for some of the early stage preclinical development work. So just moving on to talking a little bit more about multiplexing. So this is a growth area, and again, we've heard people touch on this already. So just wanted to spend a minute or two talking about multiplex immunofluorescence. So this is the ability to look at multiple targets in a single tissue section. We've been doing this work for a number of years now, and our lab over in Houston in Texas is set up in exactly the same way. We have established workflows, common processes. And we are using the OPAL system from Akoya, and this allows us to go up to interrogate 8 biomarkers in a single tissue section, and we plus a DAPI counter stain, so we can look at 9 colors with this particular technology platform. And you can see that this really kind of comes into its own when you're looking at particularly diseases where there's a high immune involvement. So you can actually, tag and identify lots of different immune cell subtypes within the tissue and look at what's happening to those from a spatial point of view, pre and post treatment, for example. So the types of data that we generate can be anything from sort of simple cell counts and looking at the intensity, for example, of certain markers that are expressed in the tissue, through to some really quite sophisticated spatial analytics. So a couple of examples just here in the smaller slides. So the one in the kind of teal and red is just an example of a nearest neighbor analysis where we are able to get the software to actually physically measure the distance between, in this case, the red cells, which are pan-CK positive, so they're our epithelial tumor cells, and CD8, so cytotoxic T cells. And within a region of interest, the software can actually physically measure the distance of those T cells versus the tumor cells, and then you can actually then get an average measurement over that region of interest, and then you can look at how that changes pre and post treatment to see whether your drug is actually stimulating those cytotoxic T cells to get into the tumor, move closer towards those tumor cells, to have their kind of autoimmune effect. The little picture in the middle here, I don't know if you guys can see my cursor or not, but it's a similar example. But here, what we've done is we've actually identified the tumor margin, and then we can use the software to actually identify regions within, so kind of banding, if you like, going from the edge of the tumor in towards the center of the tumor. And again, within each of those bands, we can actually have the software count the number of infiltrating immune cells, for example. So again, you can look at that, the real spatial changes sort of pre and post treatment. So really good way of generating high content and getting that spatial data, and it's a really good technique when you don't have a lot of material. So we're using this more and more. It's a big growth area at the moment. We're using this more and more to support exploratory clinical studies. Having said that, we also have done a lot of work with this type of technique, not only in immuno-oncology, but in other sort of immune-related diseases, so other autoimmune diseases, fibrosis, also quite a lot in skin, where being able to see that morphology and see what's going on is really important. And we've also used it a lot to characterize preclinical models. In terms of the design of these, how you can go up to 8 markers. So what markers are you going to look at? A lot of those are sort of markers for certain subsets of immune cells. And Precision has established a fairly broad range of panels that cover a lot of that sort of core immune biology. And obviously with our expertise across, both across the pond and also in our lab here in the UK, we have the opportunity to develop either completely new panels or certainly to modify panels. And so the Akoya platform is very flexible in that way, in that you can actually adapt and switch targets in and out pretty easily. We are also using the UltraView panel as well, which is a different technology for generating multiplex immunofluorescence images. So it's slightly different physical technology. It's the UltraView technology is sort of DNA barcoded based antibodies. So if anyone's interested in analysis of tissues and having a look at multiple targets actually within tissues and getting that spatial information, then do come along and have a chat to my colleague Michael, who'll be at the event on Thursday. I mentioned earlier on that we've worked with a lot of sort of weird and wacky binding molecules and things that are destined to be biotherapeutics actually, and not destined to be nice, easy to use IHC tools. So, certainly in the world of biotherapeutics, and we heard a little bit from Campbell earlier on in terms of the development of some of these biotherapeutics that Abzena can support. It's not just about monoclonal antibodies anymore. There are companies out there developing multi-specific binding molecules. There's lots of small antibody-like molecules. There's fragments, and we heard from Abzena earlier on as well about antibody drug conjugates. So, we often have to develop tissue binding assays for these, and I'm going to come on and give an example of that in a moment. But these aren't molecules that are designed to be used in IHC, really. So, you know, our expertise in assay development really comes into its fore there. So looking at you know, the ability of these tissues to bind, these molecules to bind to tissue, and also we're increasingly being asked to look at biodistribution as well. So when these molecules are administered, then actually going into the tissues with an IHC type approach and actually looking at the biodistribution of these molecules, you know, detecting them with sort of secondary antibodies to the actual therapeutic drug itself. And probably the best example of where we have to develop good binding assays for these novel biotherapeutics is for GLP tissue cross-reactivity. So this is an IND enabling technique or IND enabling assay that is required. It's on the critical path for the preclinical safety of novel biotherapeutics. And it is an IHC based technique. So basically we have to take the test biotherapeutic and we have to develop a tissue binding assay for that to screen and see whether that molecule is likely to bind off target to a panel of human tissues. And it's really there to look at, you know, is this going to cause me any safety liabilities when we go into the clinic with this? So it is IND enabling. Again, just going back to the previous slide, we've worked up binding assays for various different types of molecule. And I think probably the thing that's most important here is that it's really, really important to have a really good, reliable and robust method when you're looking at tissue cross-reactivity, because ultimately what you want is you want a negative result. You want no, or you want limited binding for your candidate biotherapeutic. You don't want it to be sticking everywhere and lighting up all the tissues. So this is an area where I think really we can exploit our experience and our expertise in working with some of these really challenging molecules and getting really good, robust IHC assays. There is a white paper that's available on our website if anyone's particularly interested in this. We'll be happy to talk to anybody that's working in this space that needs to evaluate the tissue binding for their molecules before they go into the clinic. This is a GLP assay, so we've actually been a member of the MHRA GLP compliance monitoring program for over 10 years now. I think we joined in 2010, and so data that we generate in our lab in the UK is accepted by all the regulators that form part of the mutual acceptance of data, so MHRA, EMA, FDA, the Japanese regulators, etc. So let's see how I'm doing for time. Oh, not too bad, a little bit over. So I just wanted to give one example of a cell-based assay. This was an assay that we developed from scratch to support a large pharma that were working on biosimilars when Humira came off patent. So these are anti-TNF therapeutic antibodies. And this was an assay that we, as I say, we developed from scratch. It was a fairly simple assay, 2D. We'll be using human primary cells. These were synovial fibroblasts from rheumatoid arthritis donors, because that was a good match for the biology. It's really important with cell-based assays to make sure you're recapitulating the biology correctly. And because this was going to be supporting a marketing application for biosimilars, we actually took this through the full pharmacopoeia, US pharmacopoeia guidance for cell-based assay validation. So the guys did a fantastic job, and we generated some fabulous data, and our client was able to take one of their molecules forward for marketing. And this is kind of an interesting example, because a lot of our primary cell-based assays are very customized, because it's important, as I said, to recapitulate that biology, and cell biology really isn't a case of one size fits all. So this is a really nice example where we developed an assay really for, as I say, for kind of a clinical relevance and supporting marketing application, but we're actually now routinely using it for other clients to support preclinical development for novel, both large and small anti-TNF alpha molecules. And then last but not least, in the last -8 minutes that I've got, just really wanted to mention our biospecimens piece. So obviously all the data that we generate preclinically and clinically needs to be underpinned by having access to really good biospecimens. So within Precision, we have a division that focuses on this. We have access to millions of IRB approved annotated specimens that fall broadly into these groups. I'm not going to go into too much detail, but really the message, the take home I want here is that, you know, you really need good quality samples to generate good quality data. And that's both for your preclinical research, so it could be your target and biomarker discovery and validation data. But as we think about, you know, positioning and transitioning these assays to support clinical development, you know, having really good quality tissues for your assay development and validation to support clinical assays is absolutely paramount. So with that, I know I've gone over. Thank you for your patience. Just wanted to say thanks very much. My colleague Michael Tarn will be on site on Thursday. So if you have any questions about clinical trials, specialty labs, data or sample management, please seek out Michael and he'd be more than happy to talk to you. And with that, I'd like to thank you very much.
Thank you very much, Amanda. Just as I close the session, I'd like to say another big thank you to all of our speakers today. And of course, those of you that attended, thank you for sticking with us. I appreciate we did overrun a bit slightly. Thank you for your comments and questions in the chat box. Those that didn't get picked up, we'll make sure that we follow up after the session. Yeah, it's been a fantastic opportunity to showcase what our members are up to. We're always very excited to hear it. So enjoy your lunchtime. We will see you back at 1:30 with our next innovation workshop with researcher. Thank you very much for joining.
Thank you.
Thanks, everyone.
Thank you. Bye.